So here's what I'm going to do is I'm going to get rid of all that other stuff. So hit your arrow and just make a box. Actually, I'm going to select out of this. For some reason, my box is all messed up. Dang it. Let's see if I can do it down here. I hate you. Try again. No, dang it. All right, hold on. I'll do it. I'm going to do it on the QC. You can do it that. So select those group and then hit delete. Here's what I'm going to do. I'm going to do my plot histogram. I think I can get them here. Select all those. You can just select those lines. Then over here, I'm just going to go delete. You should now have, I think, 18 samples. And you'll notice it'll highlight that stuff too. So you could actually do anything you wanted with them. Does anybody need help? What's that? So by removing those samples, what we've done is we reduce the variability that we have to account for. What's going to happen, so I want you to close your PCA plot and then reopen it. Much different now, right? This is what I want to see. In that other picture, everything was scrunched up because there was so much variability in the data set. Now that once we remove that other data set that's not even close like this one, now we've expanded that the PCA. Now we can take a look at it. What do you think the biggest difference here is? What do you think these two groups represent? Days. Yeah, totally. Let's take a look. Day. Spin this around. Yep. Absolutely. These will just go to the PCA3, but you can use PCA4. You can use as many PCAs as you want. And in fact, you can pick the PCAs you want to use. So it just gives you the first 3 that are available. But if you want to use PCA4, you could. Or 8 or 10 probably. And if you do, that's a problem. That's sometimes I hate that. Like, you never know if somebody's like manipulating the PCAs to just like visually put it so it, looks good. I always use the first 3, and then I try different combinations. And okay, so we've got day here, right? Let's select this by the. Here's what I want you to do now. Is I want you to go, instead of color, I want you to go to shape. Okay. Here's what I want you to do is I want you to put down, let's go clone. Let me get this a little bigger. Well, that don't work. Okay. So let's see. Triangles are ES cells. Squares are, is this the same for you? Yeah. This is what's cool. Like, we can tell a lot of stuff now. Look at the blue. Blue is day 0, and red is day 5. These are basically, you've got 3 replicates for each different cell type, right? So the triangles are your ES cells, your embryonic stem cells. The square one is your pluripotent stem cells from clone ST5, so it's a cell line. The other one's the same thing except from a different cell line, ST8. What do you notice at baseline? Like, if I looked at the expression of these cells, are they more similar at baseline or day 5? Baseline, totally. You can see it, right? Here's what's really cool. Look at that. You can see it. They're almost in a line. They're separate, but very similar. Now, when you start going outside of that, now you can see that this is a chain of gene expression. A bunch of genes are changing. These go way out here. What was the square ones again? ST5? I think I read in there like it actually works better. Here you have the other one, which is kind of all over the place. Again, these are pluripotent stem cells, but again, they're from a different strain, not quite like this. This one's close. But then you can see these embryonic stem cells all down here. Just by taking the data, transforming it, attaching the clinical information and experimental information from the study, we're basically able to take these samples, project them in three-dimensional space, and just based on me looking at this, I can say, okay, the stem cells, they're about the same. I mean, they're pretty similar, or at least both different kinds at baseline. Once they get induced, they all seem to behave kind of similarly. They're all going in the same direction. You could tell there's going to be a lot of similar changes. But now you're starting to see the differences. And that's where these investigators, if you read this paper, that's what they really care about, right? You know, can I use these cells, which are really easy to get, rather than these cells? So what we're going to do next time is, now that we know there's differences, let's figure out what's the difference between these different cell types at baseline and what's different as they differentiate. So we're going to do a bunch of ANOVAs. We're going to do some cool ANOVAs. We're going to do two-way ANOVAs. What you're going to do is see the statistics on that. Once we do the statistics, then we're going to start generating genes. Once we get genes, then I'm going to show you how you cluster those, how you can make heat maps. And so we're going to make all kinds of different, I'm going to show you different ways to basically display that expression data. Once we're done with that and we're settled on a gene list that we want to look at, what we will do is, now it's like, okay, we've got all these genes, what does it mean? So what we're going to do is we're going to use ingenuity pathway analysis, which is, if you had your own copy, it'd be about $10,000 per year. So you guys all owe me $10,000. So. No, they're going to let us use the software for free. So all the software, I'm still trying to get NextBio to donate their software. And if they do, all the software we're using for this class would probably cost you about $40,000 a year if you were going to buy it on your own. And so they are donating it, which is really cool. So again, what we're going to do is we're going to find out the genes that are different between these groups. And then we're going to take that and export it into ingenuity pathway analysis. And then we're going to start getting out the pathways and all that cool stuff. I'm going to tell you right now, that's my favorite part of the analysis. Is like, as soon as I put it into Ingenuity, I'm sitting there waiting for it to get done computing. I'm just like, come on, what's going on? It's just that kind of like that immediate look. And then after like 2 weeks, you're just so sick of the data set. You're like, God, I hate this data set. Stupid ES cells. All right, what time is it? About 5 minutes? Okay.